Hey, good day everyone. I'm Christoph Jung from AutoChem. Today I'm glad to introduce some of the needs and challenges of our customers in synthesis laboratories in the high-value chemical industry. I will focus on the needs of synthetic chemists that aim to develop and optimize a chemical reaction. Through AutoChem's close collaboration with leading pharmaceutical and chemical companies, we know that the R&D landscape is going through a dramatic change. Why are over 70% of the top companies heavily investing in change programs to improve R&D efficiency and output? There are three main drivers for that change. First, drug molecule complexity is increasing. Second, time to market of novel drugs is becoming more critical. And third, companies need to lower costs to stay competitive on a global market. What type of changes are required in regards to synthesis labs? On the chemistry side, complex molecules often need advanced synthesis techniques like advanced dosing and sampling, or tools that assure good results for statistical studies such as design of experiments. In addition, innovative development increasingly means the use of toxic chemistry such as high-potency reagents or wider reaction spaces such as larger temperature or pressure ranges. Time to market is strongly influenced by the team's ability to learn fast. Finding the right experimental data and sharing it with colleagues become more and more critical. As we know, many customers are still using manual tools such as the round bottom flask setup, which is often a non-standardized setup for experimentation. The lack of reliable synthesis data requires frequent repetition of experiments for data confirmation. This is a potential root cause for project delays. As cost pressure is high, scientists are expected to do more with less time and resources. They need to develop strategies to get most out of their experiments. Manual equipment, in contrast, requires them to stay in the lab, instead of focusing on value-add tasks such as experimental planning and data interpretation. There are several stages that are required to bring a molecule to the market in the high-value chemical industry. These stages are basically the same, whether we look on the biopharmaceutical or fine chemical market or CDMO type of companies. We know that across all industries, scientists follow a similar path while bringing a molecule to the market. In inventing the molecule, effective compounds must be created in the first place by chemists. Then, a reaction must be developed that offers a high-quality route to that molecule. At the same time, a process must be created that ensures that the reaction or a set of reactions are safe and robust and can be run in a batch or continuous process at larger scale. And finally, the molecules must be manufactured, both for the market, but may be equally important for clinical testing. In production, process engineers must eliminate batch failures and ultimately reduce cycle time. Something we hear from our customers is that across all areas of development, a new type of synthesis laboratory is required to make it easier to innovate and optimize at each step. When we look on the area of developing the optimal reaction route, many of our customers are asking whether the traditional setup is suitable for solving complex chemical needs and challenges. The main repetitive activities in chemical synthesis are heating, cooling, dosing reagents, stirring and taking samples for analysis. After working many years together with global customers, we have identified two main areas of potential limitations and concerns in the synthesis lab. First, the area of usability of the traditional synthesis equipment, and second, the area of collecting quality data from manual experimentations in synthesis. The usability challenge is based on the fact that manual equipment such as the round bottom flask has limitations when it comes to the manual procedures of heating, cooling, stirring, dosing and sampling. Heating with oil baths is not safe. Cooling is cumbersome and cannot cover all temperatures required for different types of reactions. Stirring can provoke inefficient mixing and insufficient mass transfer. Sampling is not reproducible and needs operator's time. Customized setups are not repeatable. The second area of concern is the limited application or condition range. For instance, by applying the typical cooling media such as ice, ice plus salt, and dry ice plus solvents, organic chemists can only cool at discrete temperature values such as 0 degrees Celsius, minus 21 degrees Celsius and minus 78 degrees Celsius. It is almost impossible to keep tea jacket at temperatures between these conventional values with manual cooling equipment. A wide application range needs to cover a wide temperature range, dosing rates, stirring speeds, catalyst rates, concentration ranges, pH value, air sensitive or oxygen sensitive reactions, pressure ranges, volume ranges, and for example, oxygen values. To solve complex chemistry problems, a simple access to a broad range of the here listed reaction parameters by using one experimental setup would be ideal. The third usability challenge is to assure personal safety at any time. The safe and error-free deployment of synthesis equipment has become a strategic imperative for many chemical and pharmaceutical companies. In regards to synthesis, the safe handling of hazardous and toxic compounds and solvents need to be assured. Besides, the equipment needs to be able to cope with unsafe situations, such as exothermic reactions. As a third aspect, modern instrumentation needs to prevent ergonomic-related injuries during frequent utilization. When it comes to quality data, traditional synthesis tools combined with manual lab notebooks can have some serious limitations. Some basic requirements for good data quality are summarized with the following three questions. Has the recipe been followed and is everything recorded? Complete recording of reaction parameter changes and analytical results is vital for process understanding. Is data retrievable? Finding, retrieving and sharing data quickly is critical to improve team productivity. Is data accurate and consistent? No transcription errors to yield reliable data, consistent data format and structure to compare datasets retrospectively is very important. The second influential factor on data quality in synthesis is the stable and precise control of reaction parameters. Let's have a look on multi-parameter experimentation with statistical analysis, for instance via design of experiments, in short DOE. In DOE, the quality of resulting statistical data describing responses such as yield and product purity depend heavily on the ability of the synthesis tool to control reaction parameters precisely within tight tolerances. It has been shown that manual tools do often fail with precise parameter control. An underestimated challenge in getting complete and quality data are information gaps. In particular, but not only for long reactions, they occur through meeting and lunch breaks or during overnight runs, for example, when no reaction samples can be taken. An unattended operation mode for synthesis control, dosing and sampling can overcome this issue and making sure that all necessary data are captured. A possible solution is a personal synthesis workstation. A personal synthesis workstation includes a reactor equipped with dosing, sampling, stirring and temperature control. This setup allows to control temperature changes precisely over the reaction period, while dosing ramps, sampling points and changes of stirrer speed can be easily programmed. Reaction data are captured automatically as the experiment proceeds, allowing the scientist to run them unattended and to have a focus on other activities. Let's take a look. First, we need to set up a recipe. In this animation, we show the programming of a heating ramp, the change of the stirrer speed to 85 RPM, a dosing ramp of reagent B for 20 minutes, a cooling phase down to 10 degrees for 10 minutes, and the stop of the experiment. Throughout the whole reaction time, we want to take a reaction sample every 10 minutes. And this is how the experimental run would look like. There is a steady dosing rate and the stirrer speed is also constant, while the heating ramp starts after a certain time and will end up at a certain programmed temperature. Then the dosing changes as it increases for 20 minutes for reagent B, while the stirrer speed has been increased to 85 and stays constant at 85. Then the cooling ramp starts at a certain time down to 10 degrees for 10 minutes. And finally, we stop the experiment. In summary, a modern synthesis setup is a simple-to-use personal workstation that delivers quality data on a day-to-day routine. Every scientist can enjoy working with it in an unattended experimentation mode with a wider application range that enables to discover new synthetic pathways in a timely manner while being protected against hazardous chemicals. The precise reaction control yields quality data which are recorded and can be easily retrieved for further data processing. At the end, I want to direct the audience here to our library of publications around the Modernize Synthesis Lab. There are many different use cases of the personal synthesis workstations, such as the development of a safe and scalable process, effective DOE studies, reaction endpoint detection, or a byproduct formation study in a pressure reaction. All these publications and references can actually be found on the website that I point to here, www.mt.com/ac-modernize synthesis. So, if you are interested in learning more how other users in industry have solved problems with the synthesis workstation, I really invite you to visit that page and download some of the publications. Thanks.